The mosquito known as Aedes aegypti originated in Africa. Over the last 400 years, it has spread around the world with increased global travel. Today, it inhabits over 120 countries in all tropical regions of the world, where it transmits the debilitating and sometimes deadly dengue virus. We're working on a way to stop the Aedes aegypti mosquito from transmitting the dengue virus. Our method uses naturally occurring bacteria called Wolbachia that lives only within insect cells, but is passed from one generation to the next through the insect's eggs. Wolbachia is found in up to 60% of all the different types of insects around us. Insects that include fruit flies, moths, dragonflies and butterflies, and some mosquitoes that bite people, but not the mosquitoes that transmit human diseases like malaria and dengue. The secret to the success of Wolbachia is how it manipulates the reproduction of the insects it lives in, so to give itself an advantage. It works like this. If a male insect has Wolbachia and mates with a female that doesn't, then the eggs she lays won't hatch. If the female has Wolbachia and the male doesn't, she would lay her normal number of eggs. They would all hatch and they will all carry Wolbachia. Similarly, mating between two insects that both carry Wolbachia will result in all eggs hatching and all offspring carrying Wolbachia. Over a few generations, the number of individuals carrying Wolbachia increases rapidly until nearly all the insects within a population have the bacteria. We had been studying Wolbachia for a long time, believing it had the potential to be used as a way to stop the spread of diseases like dengue. But to test this, we needed to be able to first move Wolbachia into the mosquitoes that transmit the virus. Not a simple task. To do this, using microscopic needles, we took Wolbachia from the fruit fly and injected it directly into young Aedes aegypti eggs. It took us thousands of attempts, but we finally did it. We had a mosquito that carried Wolbachia and would pass it to her children through her eggs. We then took the mosquitoes that contained Wolbachia, infected them with the dengue virus, and found something really exciting. The virus didn't grow well in the mosquito, and if it can't grow, it can't be passed between people. This was even more exciting when we realized that this was true not just for dengue, but for other diseases like malaria, yellow fever, and chikungunya. Our next step was to see if Wolbachia could spread into wild mosquito populations and be turned into a practical way to control human disease. We built large enclosed cages in northern Australia that we filled with local mosquitoes. We then introduced mosquitoes containing Wolbachia. Over a small number of generations, all the mosquitoes had Wolbachia. This suggested that our dengue control method had potential if we could spread Wolbachia into wild mosquito populations in communities where dengue is found. To attempt this, we needed to do two important things. First, we needed the government to look carefully at the human and environmental safety of our method and approve our trials. Then we needed to explain to people why we wanted to release mosquitoes containing Wolbachia in their communities. In 2011, with government approval and community support, we began our first open releases of Wolbachia mosquitoes in Australia to evaluate its potential to control dengue. Our hope was that our mosquitoes would breed with the local wild-type mosquitoes, and in time Wolbachia would establish in the majority of the mosquito population. We released Wolbachia mosquitoes once a week for 10 weeks, and within a few months we achieved our goal. Close to 100% of the mosquitoes in these communities had Wolbachia, and years later our monitoring tells us that they still do. The Wolbachia is maintaining itself without the need for us to do anything. We have also shown that when we take these mosquitoes into the lab, they have a greatly reduced ability to transmit dengue between people. Ongoing trials in Australia and dengue-endemic countries are now determining the best way to deploy Wolbachia across whole cities. If these large-scale trials prove as successful as our initial trials, it is our hope that the Wolbachia dengue control method will reduce the estimated 390 million human dengue infections that occur each year and help protect the health of the 2.5 billion people that live in dengue transmission areas.